Hello everyone, we are talking about cardiac septation. This is the third section of This is the third section of this lecture in which we'll be talking about the atrioventricular canals. Atrioventricular canals are the communications between the atria and the ventricles. This section has been further divided into three parts. In these parts, we'll be talking about bulboventricular flank. We'll see what that is. Then we'll talk about the valves which are present in the atrioventricular region, and finally we'll talk about the clinical correlates of as what happens if these valves or if these canals are not formed properly. So now let's just dive into it. So bulboventricular flank. I know it's a mouthful, but it's easier. It's an easy concept. I hope you'll understand it. So the fourth embryonic week is about to end. 4 atrioventricular endocardial cushions are now formed. So here, this is the AV canal, and the 4 endocardial cushions are formed, 2 on the sides, 1 dorsal and 1 ventral. So we'll see 1 on each side, 1 dorsal and 1 ventral to the AV canal. Now, bulboventricular flank is a mesenchymal area that separates the bulbous cordis. This is the bulboventricular flank, and it separates the bulbous cordis. This is the bulbous cordis from the AV canals. These are the AV canals, and this is the bulbous cordis. This is the bulboventricular flank, which is separating the bulbous cordis, which forms part of the left ventricle and the outflow tracts from the atrioventricular canals. The AV canals also give access only to the primitive left ventricle. At this point, the AV canals only accesses through the primitive left ventricle. So now the 5th week is about to end. The posterior extremity of the flank, this is the bulboventricular flank, which is separating the AV canals from the bulbous cordis. The posterior extremity of the flank terminates almost midway along the base of the dorsal endocardial cushion. So here is where the dorsal endocardial cushion might lie. So the posterior extremity of the flank almost lies midway along the base of the dorsal endocardial cushion. Now the AV canals, they begin to enlarge, and the enlargement mainly happens to the right. Now the blood passes through the AV orifice. Blood that now is passing through the AV orifice has direct access to the primitive right and the primitive left ventricles. So now let's see what the role of endocardial cushions is in all of this. Now, 2 lateral endocardial cushions were also present. This is the region of the AV canal, and the lateral endocardial cushions would be placed here. So this is an adult heart. Obviously, we are talking about a developing heart. So the dorsal and ventral cushions project further into the lumen. So they deepen further into the lumen. They fuse and divide the canal into right and left orifices by the end of the 5th week. So what do they do? They fuse with one another, and they divide the canal into a right orifice and a left orifice, and this happens by the end of the 5th week. Now let's talk about the atrioventricular valves. So these are the atrioventricular valves. Let's talk about them. So here we have each orifice is surrounded by a local proliferating mesenchymal tissue. So the AV orifice, initially this was the area of the AV orifice, which was surrounded by a local proliferating mass of tissue. Now the bloodstream hollows out the thin tissue on the ventricular surface of these proliferations. So these proliferating cells which were lined around this orifice, this AV orifice, are hollowed out on the inside by the pressure of blood. So mesenchymal tissue now becomes fibrous. This mesenchymal tissue that was surrounding the AV orifice has now becomes fibrous and forms the atrioventricular valves. Here you can see these are the valves being formed. These are actually the formed valves in an adult heart. So The valves remain attached to the ventricles by muscular cords. Initially, the valves are attached to the ventricles by muscular cords. However, with development, the muscular component of the cord degenerates and only dense connective tissue remains. So, this is how the valves are connected to the muscles of the heart. Now, the valve consists of a connective tissue which is lined by the endocardium. So, we have already talked about this. The valves are connected to thick muscular tuberculi in the wall of heart, in the walls of ventricle via the chordae tendineae. Chordae tendineae are dense fibrous tissue cords which were initially muscular, but the muscular components were lost as a result of development. The thick muscular tuberculi in the wall of heart are known as the papillary muscles. So the valves are attached to the papillary muscles via the chordae tendineae. So mitral valve, this is the mitral valve, is bicuspid. Bi means two, cuspid means cusps. So it has two cusps or two parts. So it is known as a bicuspid valve. Right AV canal, this is the right AV canal, has a tricuspid valve, that is, it has three cusps, hence the name tricuspid valve. So, heart anomalies, let's talk about those. These are the largest categories of human birth defects. They affect about 1% of live-born infants. 12% of these 1% infants have chromosomal abnormalities, and 30% may have other major birth defects as well. So here you can see various cardiac defects. This is patent foramen valve. This is coarctation of aorta. This is the patent ductus arteriosus. And this is tetralogy of Fallot. We'll be talking about all of these in detail now. First of all, let's take a look on Holt-Oram syndrome. So what is this? This is mutation in TBX gene. It is characterized by preaxial radial limb anomalies and also atrial septal defects. So atrial septal defects can be the defects in foramen veil or other ostium secundum, ostium primum, improper formation of the septum between the two atria whatsoever. The muscular portion of the interventricular septum may also be defective, and it is an autosomal dominant condition which affects males more commonly. So let's talk about hypertrophic cardiomyopathy now. So hypertrophic cardiomyopathy is caused by mutations in a number of genes which regulate the sarcomere protein production. So actually there is faulty sarcomere protein production, and it is regulated by a number of genes. So there are lot many mutations in these patients. It can cause sudden cardiac death, especially in athletes, because in this condition, the more a heart is used, the more it is at risk of sudden cardiac death. It is inherited as an autosomal dominant trait, that is, males are more commonly affected. So most mutations target the beta-myosin heavy chain gene. So this is the most common mutation, the beta-myosin heavy chain gene. So what do we see in this patient? We see myocardial disarray, which is disruption in the organization of the cardiac muscle cell. This is a figure of hypertrophic cardiomyopathy. Here you can see the thickened left ventricular heart wall. It is extremely thick, it is hypertrophic, and it is due to myocardial disarray. Moving on, we have ventricular inversion. As it's evident from the name, the ventricles are inverted in their position. So the morphological left ventricle, the morphological left ventricle is on the right side. So in a normal heart, we have right ventricle on the right side and left ventricle on the left side. In this, we have the morphological left ventricle on the right side. It connects to the atrium. It connects to the right atrium through a mitral valve. So it takes the connection between the right atrium and the left ventricle is with a mitral valve. and the morphological right ventricle is on the left side, and there's a tricuspid valve between this right ventricle and the left atrium. It is also known as L transposition of the great arteries. Why is it called that? Because the pulmonary arteries exit morphological left ventricle and aorta. Here you can see the pulmonary artery, it is exiting the morphological left ventricle. However, aorta, it is exiting the morphological right ventricle. So arteries are within their normal positions. However, the ventricles have inversed their positions, hence the name, the ventricular inversion. So what is the cause of this? It is due to the abnormality during laterality. Obviously, laterality is a condition in which during which the embryo establishes its axis, and it's very important. If there's any problems with laterality or specification of left and right sides of the heart, this ventricular inversion may take place. So now we'll talk about the hypoplastic right and left heart syndrome. So what happens? It is a rare defect in which the right or the left side of the heart is hypoplastic, that is, it has not been formed properly. So on the right side here, the right ventricle may be very small, a very small right ventricle. And the pulmonary artery may be atretic. So the pulmonary artery may be atretic or stenosed, tightened, and atrium may be small. So this is the right atrium, and it may or may not be small. It is actually small in this case. So on the left, the ventricle is very small. Here you can see a very small ventricle. Aorta may be atretic. This is the atretic aorta. Here you can see. Or stenos, and the atrium may be reduced, and the atrial size has also been reduced. It can be clearly seen in the figure. Defect in both may be due to the laterality sequences and or right-left specification of the cardiac progenitor cells did not occur during early heart morphogenesis. So again, the cause lies with the laterality or the specification of the right-left sides of the heart. Now let's talk about the atrial septal defects. Ostium secundum defect is a very significant defect. It is a large opening. This is the secundum atrial septal defect, this one. It is a large opening between the left and the right atria. It may be caused by excessive cell death and resorption of the septum primum. So as we already know, the ostium secundum is formed by the resorption and cell death in the upper portion of the septum primum. So if there's an axis of resorption and cell death in that region, it may lead to this atrial septal defect of the secundum kind, ostium secundum kind. So it may be due to inadequate development of the septum secundum, as septum secundum is the free edge of the septum secundum is supposed to cover this ostium secundum. So it may be due to faulty development of the septum secundum. Either way, depending on the size of the defect, there may be significant intracardiac shunting of blood due to this defect. So now let's talk about core trilocular biventricular. So in common terms, it is known as a common or a single atrium. So it is complete absence of the atrial septum. Here you can see an atrial septum has not been formed. It is completely devoid of an atrial septum. Hence the name common atrium. It is always associated with serious defects elsewhere in the heart. Here you can see the defect, the atrium, it's a combined large atrium with no partition in between, and it is always associated with defects in other parts of the heart. Moving on, we have premature closure of the oval foramen. So what happens is foramen oval closes during the prenatal life. It causes massive hypertrophy of the right atrium. So the right atrium enlarges in size and the ventricle as well. The left side of the heart is left underdeveloped. So death occurs soon after birth, as this condition is not it's not a viable condition, and the child cannot survive with this. So let's talk about the endocardial cushion defects. We talked about endocardial cushions. They were the extracellular matrix proliferations that were covered by cardiogenic cells. They helped in the formation of AV canals, the membranous portions of the atrial and the ventricular septums, and the partitioning of the septation in the outflow tracts. So what happens if there is a defect in the endocardial cushions? The persistent AV canal combined with a defect in the cardiac septum. So here you can see the AV canals are persistent, and there is defect in the ventricular septum as well. So septal defect has an atrial and a ventricular portion. Here you can see this is the atrial portion of the defect. This is the ventricular portion of the defects, which are separated by abnormal valve leaflets. These are the abnormal valve leaflets in a single AV orifice. Obviously, two orifices have not been formed. This is a single AV orifice, which is due to the persistent AV canal. Abnormal valves line this orifice. The defect has a ventricular component and an atrial component. So now moving on to the ostium primum defect. It is also an endocardial cushion defect. The endocardial cushion in the AV canals, now they have partially fused. So we have the primum atrial septal defect. Here you can see that it is due to partial closure of the partial fusion of the AV canals, endocardial cushions. There is defect in the atrial septum, but the interventricular septum is closed. So the interventricular septum is safe. However, there is defect in the atrial septum. There is usually a cleft in the anterior leaflet of the tricuspid valve. So that is an added finding, a cleft in the anterior leaflet of the tricuspid valve. So now let's talk about the tricuspid atresia. So there's obliteration of the right AV orifice. So the right atrioventricular orifice, it has been obliterated, as a result of which there is absence of or fusion of the tricuspid valves. So the tricuspid valve, they're either absent or fused. So it is associated with a patent foramen ovale. So foramen ovale is patent. We have a ventricular septal defect. A large ventricular septal defect can be seen in the figure. We have underdeveloped right ventricle. This area is the right ventricle. It is rudimentary or highly underdeveloped. And we have hypertrophic left ventricle. Here you can see the huge hypertrophic left ventricle. So Epstein anomaly. Epstein anomaly is a very important condition. In this, the tricuspid valve is displaced towards the apex of the right ventricle. So here, this is the normal position of the tricuspid valve. However, in Epstein anomaly, the tricuspid valve has been displaced anteriorly towards the apex of the right ventricle. There is an expanded right atrium. Here you can see a very expanded right atrium. This is the normal size of the right atrium, and this is a very expanded right atrium and a small right ventricle. The valve leaflets are abnormally positioned. Here you can see the abnormal positioning of the valve leaflets. This is the normal positioning, and this is the abnormal positioning. So the anterior leaflet is usually enlarged. Here you can see the enlarged anterior leaflet. So this was all about this section. In this section, we talked about the clinical anomalies that arise due to endocardial cushion defects and the various functions of the endocardial cushions. We saw how the endocardial cushions help in partitioning of the atrioventricular canals and the formation of outflow tracts. They also helped in the formation of the membranous components of the atrial and the ventricular septa. We saw various anomalies. All of these anomalies are very important, primarily the Epstein anomaly. They may be a part of your exams, they may be a part of your clinical training. So keep them in mind, always remember them, and for further sections, keep watching scardia.com.